Hi everyone, so today we're going to be learning about apoptosis, which is cell death. So the key knowledge from the study design is apoptosis as a regulated process of programmed cell death. What is apoptosis? Well, it is programmed cell death. This part is important, the programmed part, because programmed means that it is planned for and that it is initiated. This is something that the cell requires and it is beneficial to the cell. So it's a highly controlled process that results in the cell dying. And these are some images of some cells undergoing the process of apoptosis. So we've got some cells here. Slowly you can see them start to break down and die. So why is apoptosis important? Well, it's a crucial part of growth and development, and it has many important roles in the body, such as maintaining adult cell numbers. So we have about 50 to 70 billion cells undergo apoptosis each day. And this is to make way for new cells that are being created through mitosis. So we need to keep a balance of cells, and we can't have all these new cells being created by mitosis and not have all these old cells die. And so apoptosis is the process that these cells undergo to die to make way for new cells. Apoptosis is also important for defense against damaged or dangerous cells. For example, virus-infected cells, we don't want those cells to continue to live in us, so that our bodies will initiate apoptosis and kill these virus-infected cells, or cells with DNA damage. So if we think back to the checkpoints that we learnt about throughout the cell cycle. So if the cell picks up that there is DNA damage at one of these checkpoints, it will undergo apoptosis. So that damaged cell is destroyed and therefore doesn't replicate. Also, the transformation and sculpting of embryonic tissues during its development. So, for example, the formation of fingers and toes in a fetus. This is an image of a fetus. If you can see here, you can see that there is a webbing between the fingers. So what happens during apoptosis in this embryonic stage is that this webbing, the cells that make up this webbing undergo apoptosis, and then our fingers and our toes separate. If that does not happen and there's a lack of apoptosis, that webbing remains, like you can see in this picture. This person can live a totally normal life. It's just that at the embryonic stage there wasn't enough apoptosis that caused these cells to die and therefore separate the toes. We know that apoptosis is programmed cell death, so it is highly controlled. But why does it need to be highly controlled? Well, when cell replication and apoptosis are unbalanced, the following may occur. So if the rate of apoptosis is too high, so too many cells are dying, we can have diseases such as HIV, Huntington's disease, or Alzheimer's. For example, with Alzheimer's disease, this is when neurons, so cells in our brain, are dying too quickly, or there is too many neurons in our brains dying, and people can develop Alzheimer's. If the rate of apoptosis is too low and there's not enough cells dying, especially damaged cells, this can cause other diseases such as herpes virus or cancer, which we will get into later on. So far, we know that apoptosis is controlled or programmed cell death. So this is cell death that the body requires and that is beneficial to the cell. However, sometimes cells die unexpectedly, and this happens through a process called necrosis. So when a cell dies in an unprogrammed, unplanned way, they are undergoing necrosis. So the cell will start to swell, and it will rupture, and all the contents of the cell will spill out into the surrounding cells, causing a lot of inflammation. In comparison, apoptosis, because it's controlled, it's programmed, we don't have that spilling of the contents. The cell breaks down in a way that's very controlled into these smaller apoptotic bodies, which we'll learn about. and then these apoptotic bodies are destroyed or recycled. So apoptosis, highly controlled and planned. Necrosis, uncontrolled and causes damage or inflammation to the surrounding cells. So let's look at the process of apoptosis. There are two parts to this. First, we have initiation, and then we have execution. So I'm going to go through each of these now. Apoptosis. We'll start with initiation, so how apoptosis begins. So it can be initiated through two pathways. The mitochondrial pathway, which is intrinsic, so internal, so it's initiated internally, or the death receptor pathway, which is extrinsic, or the apoptosis is initiated externally. The death receptor pathway can also be called the death ligand pathway. You can also call this the extrinsic pathway, and so the mitochondrial pathway can also be called the intrinsic pathway. Summary image of the extrinsic and intrinsic pathways. There looks like there is a lot going on, but I'm going to step you through each of these steps. Don't be worried about all these different terms and remembering all of this. I'm going to tell you exactly what you need to remember. This just gives you a very clear idea of the process. So let's go through the mitochondrial pathway, also the intrinsic pathway. The mitochondria receives an internal stress signal from the cell. So if we've got the mitochondria inside the cell, the stress signal is coming from inside the cell. This might be because DNA is damaged by toxins or radiation or whatever, but that signal or that damage is being picked up by the mitochondria. Then we have proteins these proteins here, you don't need to know the names, but they help or they perforate the mitochondrial membrane, meaning that the membrane becomes more permeable, and this allows a molecule called cytochrome C, these orange ones here, to leak out into the cytosol. This cytochrome C activates a whole heap of different proteins. So the apoptosome, then procaspase 9, then caspase 9, then caspase 3. We call this the caspase cascade. So one caspase enzyme is activating another caspase enzyme, is activating another caspase enzyme. If you don't know what an enzyme is, it is essentially a protein that helps speed up a chemical reaction. So we've got damage being picked up by the mitochondria. These proteins perforate this mitochondrial membrane, so they create holes in it, make it more permeable. A molecule called cytochrome C, I want you to know this term, cytochrome C, it leaks out into the cytosol, which then initiates a caspase cascade of enzymes. This caspase cascade, or these caspase enzymes, activate other caspase enzymes, and then which leads to the controlled breakdown of the cells. So DNA is degraded and the cytoskeleton and organelles start to break down. So apoptosis has begun. Apoptosis is initiated. You can also have apoptosis initiated through the death receptor pathway or the extrinsic pathway. So the signal is coming from an external source. So a death signal molecule, like this one here, binds to a death receptor on the surface of the cell. So it is a protein that's on the surface of the cell that a death signal molecule will bind to. This is really called a death receptor, and it really is on our cells. So the molecule binds to the death receptor, and this then activates caspase, or proteins then that activate caspases. So again, there's a caspase cascade of enzymes, which then starts to break down the cell through DNA being degraded, the cytoskeleton and organelles breaking down, and then we've initiated apoptosis. So here's this image again, this summary image. I want to point out the terms that I want you to know, because there is a lot of different and new words on this image, so I want to point out the words to you that I want you to know. So I want you to know about the death receptor. So in the extrinsic or death receptor pathway, there is a death receptor on the surface of our cells that a molecule binds to initiate the extrinsic pathway. I want you to know that there is a caspase cascade. I don't need you to know the different numbers or names of the caspase enzymes. I just need you to know there's a caspase cascade. For the intrinsic pathway, I want you to know that cytochrome C is released once the mitochondria picks up the internal stress signal. Right? So I want you to know the name of this molecule, cytochrome C. And then again, I want you to know that there is a caspase cascade. Now that apoptosis has been initiated, the second stage is execution. So we have initiation and now execution of apoptosis. So the cell starts to shrink as it's broken down by these caspase enzymes. The plasma membrane then starts to form bubbles called blebs. And this is one of my favorite words in biology. It really is a bleb, so it's kind of just a bubble on the surface of the cell. The chromatin, so that genetic material, starts to condense and the nuclear membrane starts to fragment and break down. And the cell starts to break into these smaller membrane-bound portions called apoptotic bodies. And so inside these apoptotic bodies is densely packed cellular contents. So nothing is spilling out, it's all very contained in these small little apoptotic bodies. Apoptosis done. So once we've got these apoptotic bodies and apoptosis has occurred, what then happens is we have to remove these apoptotic bodies. What do we do with them? So phagocytes, immune cells, are attracted to areas where apoptosis is happening. And these phagocytes engulf the apoptotic bodies to destroy the cellular material. So if you think back to when we did transport of substances across the plasma membrane, we learned about endocytosis and exocytosis as well as phagocytosis, which is the engulfing of foreign bodies or physical material by cells. So here we have a phagocyte type of immune cell engulfing the apoptotic bodies, and they start to degrade it and destroy it. And so we no longer need this cell, or the cell is damaged, we cannot use it, so we need to destroy it. What I have here is a video which I can't play right now through this video, so I've linked it in the description below, but we'll also be watching this in class, and it's just an animation of the process of apoptosis. So it's linked below if you want to watch it, but we will also watch it in class. So like all biological processes, sometimes malfunctions occur. So one of the malfunctions in apoptosis is that not enough apoptosis is happening. And we know that cells can detect incorrect DNA replication or mutations or cellular damage at the cell cycle checkpoints, so G1, G2, and metaphase. And then the cell will initiate apoptosis. But if the cell doesn't pick up on this, on these mutations or this damage, the cell with the damaged DNA will continue to replicate, and this will lead to tumor growth. This is what we call tumors. So not enough apoptosis means too much cell growth, which also means tumors. Too many cells being created will result in tumors. So some cells may also have a faulty death signal receptor. So if you think back to the extrinsic pathway, the death receptors on the surface of the cell, if that is faulty, it can also prevent the initiation of apoptosis when those death signal molecules are released. And this is common for cancerous cells, and this is how cancerous cells survive, because there is a faulty death receptor, and therefore apoptosis does not occur. And then we have tumor growth. So what are tumors? We were saying that not enough apoptosis will result in tumors. And so tumors are abnormal cells that are replicating rapidly. They can either be benign or malignant. So if we have a benign tumor, it's enclosed in a capsule and doesn't spread and invade other parts of the body. Sometimes when we have a benign tumor, it doesn't need to be removed from our body because it's not causing us any harm. However, we usually monitor this and detect it for any changes. So this is kind of what a benign tumor will look like. It's enclosed in this capsule. It's not spreading out into these normal cells. These cells still might be replicating pretty rapidly. And if it grows too big or it starts to spread into normal cells, that's when we would consider it a malignant tumor or cancer. So this spreads into other parts of our body and creates even more DNA mutations. And this can affect the functioning of the tissue that it starts to invade. So cancer is a disease caused by uncontrolled replication of damaged cells that can spread to other parts of the body. This is kind of what it looks like. We've got those damaged tumor cells spreading and causing more damage in the body. So these cells aren't undergoing apoptosis. And so the problem here is not enough apoptosis leads to cells replicating rapidly and then causing damage, which is what we call tumors. Like I said, not enough apoptosis can lead to tumors, which we consider to be cancerous if they're malignant, if they're spreading to healthy cells or healthy parts of our bodies. So mutations to certain genes, that means DNA damage. When the genes or this DNA damage occurs in genes that control cell replication or the genes that control apoptosis are damaged or there are mutations in them, this can lead to uncontrolled cell division and result in the form of cancer cells. So for example, we have something called a P53 gene that's associated with many types of cancer. So the P53 gene produces a P53 protein, and this is involved with initiating extrinsic apoptosis pathways. If we have a mutation or damage to this specific P53 gene, that means the initiation of apoptosis does not occur, and therefore we can have cancer. This is an image of some kidneys. You could see down here there's some healthy kidney tissue, but for the most part, this yellowy brown part is cancerous tissue, and so this is tumors. This is uncontrolled cell division because not enough apoptosis is happening, and this might be a result of a mutation to the P53 gene. A lot about what happens if there's not enough apoptosis, and that can lead to cancer. However, if we have too much apoptosis, that can also cause disease. For example, when apoptosis is initiated too much and more cells undergo programmed cell death than is required, this can lead to diseases such as Alzheimer's, like I mentioned before. So we might have high levels of these specific enzymes which promote intrinsic apoptosis. And so you have a lot of cells in your brain dying, therefore causing Alzheimer's. Other viruses can stimulate apoptosis, such as HIV, which stimulates apoptosis and accelerates the destruction of T helper cells, so immune cells. So too much apoptosis is happening as a result of the HIV, which is a virus, causing disease. So here we have just a really cute cartoon describing apoptosis. So we know that apoptosis is programmed cell death, so we want to eliminate unwanted cells. It's beneficial to do this. Cells start to shrink after it is initiated, either through the intrinsic or extrinsic pathways. The nucleus starts to fragment and the organelles start to break down. The membrane blebs and we form apoptotic bodies, which is then engulfed by phagocytes. So we have the phagocytosis of apoptotic bodies. They are destroyed, there is no inflammation, there is no harm to other cells. It is programmed, it is planned, it is highly controlled. So I'd like you guys to write down any questions you have and bring them to class as usual, and I will see you there.